Inflammation. This presentation is intended to summarize and simplify the complicated process of inflammation. It outlines the pathogenesis, or the development, of inflammation in living tissue. Some type of an injury initiates the process of inflammation. This could be a toxic chemical, a physical force, an infectious microbe. The body's first response is through changes in circulation. It begins with a transient or brief constriction of the arterioles and the smooth muscle cells, followed by vasodilation or relaxation. As a result, arterial blood rushes into inflamed tissues, which is referred to as hyperemia, or too much blood. Increased pressure from all of this blood forces plasma through the vessel walls, which causes edema or swelling. Meanwhile, because vessels expand, their blood flow slows down, which leads to congestion. This congestion triggers hemodynamic changes, which are changes in the distribution of the different blood cells. Red blood cells form stacks called rouleaux. White blood cells also migrate toward the margins of the vessel's interior space in a process called pavementing. These circulatory changes lead to hyperemia. Note the contrast between the normal capillary network and the capillary network experiencing acute inflammation. An injury triggers a nervous impulse that relaxes the precapillary sphincters in the arterioles, which allows blood to flood the capillary network. This slide focuses upon the cellular changes in inflammation. You can see the stacking or rouleau of red blood cells. Step 1 is the margination of a type of white blood cell called neutrophils, which bring them in close contact with the vessel's endothelium. Step 2 is the adhesion of platelets, which results in the release of chemical mediators of inflammation and the coagulation of blood. Strands of fibrin form, which are the first of clot formation. Step 3 involves the pavementing of neutrophils, which is mediated by adhesion molecules activated by mediators of inflammation. Here we focus upon changes in the vessel's walls and increased permeability and lead to edema or swelling. These include A. Increased intracapillary pressure. B. Slowed blood flow. C. The adhesion of leukocytes and platelets to endothelial cells. D. The action of soluble mediators of inflammation found in the plasma. And E. The action of mediators of inflammation which are released from special inflammatory cells. What are these chemical mediators of inflammation? Well, they can be divided into two major groups: the plasma-derived or the cell-derived. They are biochemically diverse and include biogenic amines, such as histamine, peptides, such as bradykinin, and arachidonic acid derivatives. These mediators are multifunctional and may be involved in several inflammatory reactions, act on many different cells and tissues in the body, and many of them have overlapping functions. Let's differentiate between the cell and plasma-derived mediators of inflammation. One of the most important cell-derived mediators is histamine. It is released from platelets and mast cells. It stimulates a retraction of the vessel's endothelial cells. This creates gaps between the cells and results in increased permeability. Histamine acts and therefore initiates an immediate transient reaction. Histamine, this is in contrast to the mediator bradykinin. Bradykinin must be activated by a protein in the plasma called the Hagemann factor. The Hagemann factor also acts to coagulate and clot the blood. Bradykinin is similar to histamine in that it promotes vascular permeability, but because it has to form in the plasma, it acts more slowly. Unlike histamine, bradykinin also induces pain. This slide summarizes what happens when Hagemann factor is activated. It shows the three end products, bradykinin, plasmin, and fibrin, and their functions. Besides bradykinin, the activation of Hagemann factor leads to the synthesis of plasmin and fibrin. Fibrin is involved in clotting. Plasmin helps to break apart fibrin clots and also stimulates more Hagemann factor, thereby sustaining the inflammatory response. Finally, plasmin also activates another important source of mediators found in the complement system. What are the main features of the complement system? Well, they are a group of plasma proteins produced by the liver, found circulating in the blood in an inactive form. These series of proteins are activated through the classical, alternative, or lectin pathways. Activation occurs as one protein acts upon the next in a complement cascade. During the course cascade, various intermediate complexes and fragments, all of which are biologically active, are formed. But this cascade terminates in the formation of the membrane attack complex, or MAC for short, which is able to destroy the target cells with enzymes that bore holes in their membranes, promoting cell lysis. This slide illustrates three different pathways for complement activation. The classical pathway is activated by antibodies. The alternative by substances like bacterial and fungal toxins, or snake venom, and the lectin by the protein lectin that binds to the surface of bacteria. Along the way, many intermediate derivatives form that serve different functions. But again, all three pathways converge upon the formation of the membrane attack complex, which causes cell lysis. These intermediate complement derivatives have three main functions, as shown in the previous slide. First is opsonization. Complement fragments called opsonins bind to target bacteria. This allows the white blood cells to recognize them as pathogens, so that they become engulfed in the process of phagocytosis. The second is anaphylaxis. Complement called anaphylatoxins stimulate the release of histamine, which increases vessel permeability and promotes vasodilation. The third is chemotaxis, where complement serves to attract white blood cells to the site of inflammation. Finally, there is an important group of mediators that come from arachidonic acid, an omega-6 fatty acid derived from cell membranes through the action of white blood cells. Arachidonic acid is metabolized along two pathways, the lipoxygenase pathway and the cyclooxygenase pathway. The lipoxygenase pathway produces leukotrienes. These promote chemotaxis, vascular permeability, and the bronchospasm associated with asthma. And it produces lipoxins. These oppose the effects of leukotrienes and are therefore considered anti-inflammatory. The cyclooxygenase pathway produces thromboxane. This promotes platelet aggregation and thrombosis. It also produces prostacyclin, which opposes the effect of thromboxane. And it produces prostaglandins. These are involved in smooth muscle contraction and relaxation. They also mediate pain and fever. This last slide illustrates these two pathways from the metabolism of arachidonic acid. One is the lipoxygenase pathway, which terminates in the production of leukotrienes and lipoxins. The other is the cyclooxygenase pathway, which terminates in the production of thromboxane, prostacyclin, and prostaglandins.